Hello everyone. Today my topic is about cytokines. So let's go to the session. So cytokines, often referred to as the messages of the immune system, or a diverse group of small proteins secreted by various cells throughout the body. These signaling molecules act as sophisticated communication network, orchestrating a wide range of response within the immune system and beyond. They are like conductors of an orchestra directing different immune cells to perform their roles in maintaining health, responding to challenges, and ensuring body's overall well-being. Cytokines are pivotal in regulating inflammation, immune defense, and tissue repair, and even influencing physiological processes far beyond the immune system scopes. Then how cytokines are signaling? There are three processes. One is autocrine, endocrine, and paracrine. Our cytokines have an autocrine effect when they act on the same cells that produce them. In other words, they influence their own producer cells, often amplifying a particular response within that cells. Endocrines. The cytokine function in an endocrine manner when they are released into the bloodstream and travel to distant target cells or tissues to exert their efforts. This is similar to how hormones work. Paracrines. Cytokines act in a paracrine fashion when they affect the neighboring cells in the immediate vicinity of the producing cells, playing a role in localized cell-to-cell communication within the tissue. Let's see about the properties of cytokines. First one is pleiotropic. This refers to the ability of a single cytokine to have multiple and diverse effects on various cell types or processes in the body. Reductancy. In the context of cytokine, the reductancy means that multiple cytokines have similar or overlapping function. So if one is missing or ineffective, others can compensate to maintain a particular response or function. Next is synergy. Synergy occurs when two or more cytokines work together in a way that enhances or amplifies their combined effect, producing a more significant response than each could achieve individually. Antagonism. This is when one cytokine inhibits or counteracts the effects of another, often to fine-tune or balance immune response or other physiological process. Next is the types of cytokines. There are five types: interleukins, interferons, tumor necrosis factor, colony-stimulating factor, and chemokines. Let's see one by one. So first one is interleukins. Interleukins are a group of cytokines that specifically regulate communication between with white blood cells. They play a central role in orchestrating immune cells by promoting cell growth, differentiation, activation, as well as modulating inflammation. Interleukins are numbered and have diverse functions contributing to the body's defense against infection, tissue repair, and regulation of various immune-related processes. Next is interferon. Interferons are a group of signaling proteins produced by cells in response to viral infections and other immune challenges. They play a critical role in antiviral defense by inhibiting viral replication, enhancing immune response, and regulating cell growth and differentiation. Interferons are an important component of the body's innate immune system and are regulated and are used therapeutically to treat certain viral infections and diseases, including hepatitis and multiple sclerosis. Next is tumor necrosis factor. Tumor necrosis factor is a cytokine that plays a key role in inflammation and immune response. It can promote inflammation and cell death and is involved in the body's defense against infection and the regulation of certain immune processes. TNF is also implicated in various inflammatory diseases and medications targeting TNF are used to treat conditions like rheumatoid arthritis and inflammatory bowel disease. Colony stimulating factor are cytokines that promote growth, development, differentiation of white blood cells, particularly granulocytes and macrophages. They play a vital role in regulating the production and function of these immune cells, contributing to the body's defense against infection and the recovery from the illness or injuries. They are used clinical to treat conditions characterized by low white blood cells counts, such as the chemotherapy-induced neutropenia. Next is chemokines. Chemokines are a family of small signaling proteins that play a critical role in immune response and inflammation. They primarily function as chemoattractants, guiding immune cells to the site of infection, injury, or inflammation in the body. Chemokines are essential for cell migration and positioning within the tissues and are critical in regulating the recruitment and activation of immune cells such as leukocytes. They contribute to the body's ability to mount an effective immune response and maintain tissue homeostasis. Let's see about the functions of cytokines. This is the overall cytokines which are doing their functions in each cell. Let's see. Let's categorize the cytokines based on their functions. First one is pro-inflammatory cytokines. These cytokines are involved in promoting inflammation, immune response, and host defense. Interleukin examples are interleukins 1, 6, 8, 12, 17. Tumor necrosis factor alpha and beta. Chemokines IL-8, CXL-CL8, CXCL1, CXCL2. Anti-inflammatory cytokines. They are like suppression of immune inflammation. These cytokines help to regulate and dampen the excessive inflammation. Interleukin 4, 10, 13, 31. Tumor necrosis factor non-specifically. TNF-alpha can have both pro-inflammatory and anti-inflammatory effects depends on the context. Interferon beta, lambda, and maintain homeostasis. Next is immune cell activation and differentiation. They play a role in activation, differentiation, and proliferation of immune cells. Interleukin 2, 4, 12, 15, 12. Interferons alpha. Interleukin gamma, colony stimulating factor GCSF and GM-CSF. Chemokines CCL19, CCL21. Hematopoiesis. Cytokines involved in the regulation of hematopoiesis, including erythropoiesis and thrombopoiesis. Interleukin 3 and 7. Colony stimulating factor M. Macrophage colony stimulating factor erythropoietin and thrombopoietin. Next, antiviral response. The cytokines that contribute to the body's defense against the viral infections, interferon alpha, beta, and gamma. Autoimmune and inflammatory disease. Cytokines that are implicated in autoimmune disease and chronic inflammation. Interleukins 23 and 17. Tumor necrosis factor alpha. Chemokines CCL20. Tissue repair and fibrosis. Cytokines involved in wound healing and tissue remodeling. Interleukin 6 and 10. Tumor necrosis factor alpha. Colony stimulating factor GM-CSF. Next is chemotaxis. Cytokines that guide immune cells to a specific site of infection or injury. Chemokines CXCL12, CXCL10, CCL5. Lymphoid organ development. Cytokines are crucial for development and organization of lymphoid tissues. Interleukin 7 and 2. Tumor necrosis factor beta. Chemokines CCL19, CCL21. Immune suppression. Cytokines that inhibit immune response and promote tolerance. Interleukin 10 and 37, 35. Interferon gamma. So next see about the JAK-STAT pathway. So what is this JAK-STAT pathway means? It's primarily signals the cells activate or regulate gene expression in response to cytokine stimulation. It doesn't directly produce cytokines itself, but rather it serves as a key interstellar signaling pathway through which cytokines induced signals are transmitted from the cell surface to the nucleus. Here is how the JAK-STAT pathway works. Cytokine bind. First is cytokine binding. It binds with the It begins with the binding of cytokines to its specific cell surface receptor. These receptors are often associated with receptors associated with Janus kinase. Next is JAK activation. The binding of cytokine to its receptor induces conformational change in the receptor, bringing two associated JAKs into close proximity. Proximity allows JAK to phosphorylate each other and the receptor. STAT recruitment and activation. Phosphorylated receptor tyrokinase residues create docking site for signal transducer and activator of transcription. That is the STAT protein. STATs when recruited and themselves are phosphorylated by JAKs. Next is STAT dimerization and translocation. Phosphorylated STATs off from the dimers and then relocate from the cytoplasm to the nucleus. Next is gene regulation. In the nucleus, STAT dimers bind to the specific DNA sequence known as STAT responsive elements within the promoters of target genes. This binding results in the activation or repression of gene transcription. So, thank you for listening. Thank you.